Hello guys, I am Shubham from ShubhVed Tutorials. This video is about transformation. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. So transformation is the transfer of genetic material. Okay, same as we saw in the conjugation. It is a method of transfer of genetic material, but the genetic material is extracellular here. Okay, and it is also a unidirectional one-way transfer. Okay, so unidirectional transfer of extracellular genetic material occurs in transformation, right? In the conjugation previous video, we have saw the genetic material was present inside the bacterial cell, that is the plasmid. Okay, we called it as F factor. It was intracellular. In this transformation, the genetic material is extracellular, right? So this occurs in two types transformation: natural and artificial. So these are the two types of transformation. In natural, the good example is bacillus. Okay, bacillus species of bacteria. And in artificial transformation, good example is E. coli. Okay, E. coli. To enhance the transformation, okay, efficiency of transformation, cells are treated with chemically. Okay, different chemicals, right? Or electroporation. So electroporation is very important. Mostly electroporation is used to enhance efficiency of transformation. What we use? Chemical treatment or electroporation. Electroporation. Okay. So what is this electroporation? Electroporation means electric field is applied on the cells, and poration means pores are formed inside the cells. Okay, and then the cell become more permeable to this transfer of genetic material. Okay, so this technique, chemical treatment or electroporation, is to make cells more permeable for the entry of genetic material, right? Okay, in natural transformation, transformation naturally occurs naturally, and in artificial what we do, artificial transformation, here see donor and the recipient. So we do extract DNA, extract, purify, and break into small fragments. Break into fragments what? DNA, right? We extract DNA, purify it, and break it into fragments. And the recipient, we do electroporation to make the cell more permeable for this entry of DNA, right? Electroporation, and or we can say chemical treatment. Chemical treatment, right? After this, the recipient is called as competent cell. Okay. competent cell because it will compete for the with the donor. It will compete with the donor. Recipient is known as competent cell. After we make it efficient for the transformation, right? We are making it more permeable so that DNA can enter into it, right? So this is what we do in the artificial transformation. How this occurs exactly, we will see with the help of pictures. So see, in this picture, this is a recipient, okay, recipient DNA, and this is a double strand donor DNA. Donor DNA is A plus. See, this is A plus, and here it is small a. Okay, we can say these are homologous to each other, right? So during entry, what happens? Only one strand comes in, other strand is degraded. Okay, only one strand is entered, other is degraded. Then it will bind to the double strand DNA. Okay, and this is known as triple stranded structure, right? Triple stranded structure. So after this, there is recombination by double crossover. See, double crossover is taking place, and then this A plus is getting entered into the bacterial chromosome, and from the bacterial chromosome, only A, the homologous, these are the homologous to each other, getting exchanged, right? Then this part, exchanged part is degraded. So the next important term is heteroduplex DNA. Heteroduplex DNA. See, when this type of chromosome is there, in which one strand is having a different gene and another strand is having a different gene, right? A and A plus. Then this is known as heteroduplex DNA. Okay, from the double stranded DNA, one strand is giving different information. Genetic information is different on one strand, and on another strand, the genetic information is different, or we can say genes are different. Okay, as we are seeing here, on one strand it is A, and on another strand it is A plus. So after application of recipient chromosome, what happens? One cell will be with A plus, right? A plus, and another will be with A plus, only A. So this is known as non-transformant. See, non-transformant because here transformation is not occurring. There are no transformation genes here, and here there are transformation genes. So this is A plus transformant, right? So this is how transformation take place. And what is the application of transformation now? Why we do these things? See, first of all these techniques, conjugation, transformation, and transduction are used for genetic mapping in bacteria, right? Genetic mapping in bacteria. So we can understand the gene order. Okay, gene order we can understand, right? Gene order by co-transformation. Okay, so what is this co-transformation now? See, co-transformation is two genes are getting transferred at the same time. Okay, two genes. So those two genes, when they will get transferred, when they are close to each other, right? When they are close to each other. See, in this example is donor DNA. See, it is having three genes: P plus, Q plus, O plus. Okay, see, extract DNA from population of donor bacteria. DNA breaks into fragments, right? Okay, then transformation genotypes. So what we can see, these are the fragments. Okay, and DNA fragment used to transform PQ recipient. Recipient will be PQO. Now first transformation genotype is P plus QO, then P plus Q plus O, then P Q plus O plus, and here PQO plus. So what we can understand, P plus is going with Q plus. So these are close to each other, and Q plus is going with O plus. Means the order is PQO. Like this we can understand. See here, P and Q are located close to each other. Q and O are located close to each other. So what is the order? P, Q, O. Right? So this is the order. Like this we can understand the gene order. Okay, when the co-transformation is seen. Co-transformation means two genes are getting transferred at a single time, right? So this is all about transformation. So I think this video helps you. If you find this video helpful, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever the video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.